Hi folks, welcome to my channel, The Average Dermadoc. Let's begin the show. Type 4 hypersensitivity reaction. It is entirely different from the remaining three hypersensitivity reactions where they are immediate and humoral antibody mediated. Here it is delayed. How much delayed? 2 to 3 days. Cell mediated. Which cell? T cell. It is given the name delayed type hypersensitivity T cell, where tissue damage occurs predominantly due to activated macrophages. Here it has similarity with type 1 reaction as it has two phases of mechanisms. One is sensitization phase and effector phase, same as type 1, but the difference is here it is macrophage mediated, type 1 is mast cell mediated. Same type 2 and type 3 also has similarities. Both are antibody mediated. Type 2 is only antibody will go and attack the target cell, whereas in type 3 both immune complexes, means antigen and antibody combination, immune complex will go and attack the tissue, own tissue. Coming to sensitization phase, we already know how sensitization occurs in type 1 hypersensitivity reaction. Same here, antigen is presented. First antigen will be engulfed by antigen presenting cell and then processed to present the antigenic peptide, not the whole antigen, just a piece of antigen called antigenic peptide. That piece will be a proteinaceous material of antigen. How it will present? Via MHC class 2 to T cells. How T cells will accept? It has MHC class receptors to accept, nothing but T helper cells. This delayed type of T helper cell will be derived from activated T helper 1 cell, majority mainly, and CD4 plus cells, CD8 plus cells, T helper 17 cells. These also can be converted to delayed type hypersensitivity T cell, but majority type 1 helper cell. Whereas in type 1 hypersensitivity it is T helper 2 cell, here T helper 1 cell, which gets differentiated into delayed type hypersensitivity T cell. Now our effector cell, delayed type T helper cell arrived. It will be waiting for subsequent antigen exposure in effector phase. On subsequent exposure to same antigen, this delayed type hypersensitivity T helper cell secretes various cytokines which attracts, recruits, activates various inflammatory cells, mainly macrophages, at the target sites, releasing lytic enzymes. This macrophages releases various lytic enzymes which leads to tissue damage, ultimately leading to delayed type 4 hypersensitivity reaction. What are those cytokines which is activating macrophages? After activating, what macrophage will do? We will see now. Here our effector cell, delayed type hypersensitivity T cell, is releasing cytokines like interleukin 2. First it will act on delayed type T helper cells like T helper 1 cell, CD4 plus, CD8 plus cells in an autocrine manner to stimulate the proliferation of delayed type T helper cells. First it will release interleukin 2, which will help for the utilization of itself in an autocrine manner. Then interleukin 3 and granulocyte monocyte colony stimulating factor, which helps in local synthesis of monocytes. Monocytes will go to tissues and gets converted to tissue macrophages. Here then tumor necrosis factor beta. It will help in monocyte migration and then it will get into tissues. There it is called tissue macrophage, which causes tissue damage. Then main interferon gamma activates resting macrophages, which leads to granuloma formation. Remember like this: main interleukin, interferon gamma, G for gamma, G for granuloma formation. Continuous delayed type hypersensitivity reaction. Why it will occur continuously? Because of intracellular microbes which are persistent and non-degradable, unable to kill. Then continuously it will try, delayed type hypersensitivity cells will try to kill them by activating macrophages. Macrophages release lytic enzymes. Usually this macrophages delayed type hypersensitivity will eliminate in normal individuals, whereas in individuals with persistent and non-degradable intracellular pathogens, it is unable to eliminate, so the macrophage will release more and more lytic enzymes, causing own tissue damage. And granulomas will be formed. What is a granuloma? What it consists? It consists an inner zone of epithelioid cells surrounded by collar of lymphocytes, nothing but our T helper cells, and peripheral rim of fibroblasts and connective tissue. These granulomas are formed to contain the intracellular pathogens to one site. Example: tuberculosis, trematode infections, schistosomiasis, sarcoidosis, leprosy, type 1 lepra reaction. In all these, granulomas will be formed, which contains epithelioid cells. What are epithelioid cells? Epithelioid cells and multinucleated giant cells are formed by our activated macrophages. Initial T helper cells infiltrate will be progressively replaced. Progressively means slowly, means it takes time, 2 to 3 weeks, and then macrophages will come and replace this T helper cells, which then transforms into two types of cells, nothing but our epithelioid cells and multinucleated giant cells. What is an epithelioid cell? Macrophage becomes large, flat, eosinophilic cell called epithelioid cell. These epithelioid cells occasionally fuse, induced by interferon gamma. I already told you, G for gamma, G for granuloma, to form multinucleated giant cells. Fused epithelioid cells which contain multiple nucleus and a single cytoplasm, large fused cytoplasm, that is nothing but multinucleated giant cell. Now coming to examples of type 4 hypersensitivity reaction. Main contact dermatitis. In response to contact antigens such as nickel, poison ivy, poison oak, picryl chloride, most of these antigens or substances are haptens, which means they don't have immunogenic property, which means they won't induce immunity in the host. They will combine with host proteins like albumin, globulin. Here in skin, it combines with skin proteins. Then this complex will be immunogenic. After combining with any protein, it will become immunogenic, means it induces immune system in the host. These skin proteins act as carriers and make the hapten immunogenic, and then presented by antigen presenting cells. In skin, what are those antigen presenting cells? Langerhans cells to T helper cells. Then T helper cell converts to delayed type hypersensitivity T helper cells and inducing delayed type hypersensitivity reaction through activating macrophages, which release lytic enzymes, resulting in skin lesions, redness, pustules. Now other examples: skin tests like tuberculin test. It is the prototype example of delayed type hypersensitivity reaction. Here tuberculin antigen will be injected intradermally and we will wait for some time. How much time? 2 to 3 days, because it is delayed type reaction, so it will take time, 2 to 3 days. We will observe induration and erythema, swelling at the site of injection. Why? Because if the host is already sensitized and ready with delayed type hypersensitivity T cells, then he will show the reaction and subsequent exposure to this tuberculin antigen. Same lepramin test, Montenegro test for leishmaniasis, Fry test for LGV. These are all examples of delayed type hypersensitivity reaction. Intracellular pathogens like bacteria, TB, leprosy, fungi, candida, cryptococcus, histoplasma, pneumocystis jirovecii. Non-infectious conditions, type 1 diabetes mellitus, multiple sclerosis, Crohn's, chronic transplant rejections, graft-versus-host disease. I already told you, wherever the graft word comes, we have to answer type 4 hypersensitivity reaction. Here I mentioned clearly chronic transplant rejection, because in hyperacute transplant rejection, major role will be played by type 2 hypersensitivity reaction, means antibody-mediated. We will revise the entire thing with the help of this diagram. First, antigen will be presented by antigen presenting cell. This is the antigen presenting cell through MHC class 2 to T helper cell. This is our T helper cell. On presentation by antigenic peptide by antigen presenting cell, it will get activated and gets converted to delayed type hypersensitivity T cell. Here it is T helper 1 majority, I already told you. It will release cytokines. Here black dots are cytokines like interleukin 2, which acts in autocrine manner. See, here it is acting on its own cell to stimulate and synthesize more and more delayed type T helper cells. Now delayed type T helper cells will be ready for the effect. Until here sensitization finished. Now on subsequent exposure to the same antigen, this delayed type T helper cells releases various cytokines: tumor necrosis factor beta, interferon gamma. Main interferon gamma will convert resting macrophage to activated macrophage. Resting macrophage, here it is containing some MHC class 2 to present antigens, and this blue ribbon like structure, tumor necrosis factor receptors. Here after activation, more and more tumor necrosis factor receptors, more and more MHC class 2 receptors, more and more oxygen radicals and nitric oxide will be there to attack the intracellular pathogen. Instead, it will attack and causes own host tissue damage. This activated macrophage is the main reason of delayed type T helper cell reaction. This is effector phase. Remember the channel name, The Average Dermadoc. Don't forget to like, share and subscribe. Thank you. Hasta la vista. До свидания.